everyone, my name is Camila Rodriguez and today I'm going to reveal and explain the genetic challenge A recuperation for biology. So part 1 is multiple choice questions. So first, in a cross of AABBCC multiplied by the same AABBCC, what is the probability of producing the genotype AABBCC all dominant? As you can see here, in this problem, we have a lot of data. So the first step, it will be to collect all the data that we have. For the first part, we have that all of them, we have to collect first what are we going to use to solve the exercise. For example, in this part, we are going to notice all the alleles that are disposable for our solution. In this case, heterozygous AA, heterozygous BB, and heterozygous CC for both of the cases. And the exercise is asking for a probability. So the probability can be seen as 1/2 of chances, 1/4, or 1/8. So we need to focus on the answer that is asking that all of the traits, the probability to get all of the traits dominant. So now we can proceed with the explanation of the answer. For this exercise, we need to use the Punnett square. Why? Because like this, you are going to be able to see the alleles with the crossing over to get the genotype. So in this case, we need to organize the dominant, recessive, and then solve the Punnett square, which will show us one dominant gene, heterozygous for A dominant and A recessive, the same over here, and then a complete recessive trait, a complete recessive gene. Can you see the pattern in here? Since we have heterozygous, one dominant and one recessive, or one capital letter and one lowercase, we are going to have always the same, which is one dominant, two with one dominant and one recessive trait, and in the last one we will have a recessive. So this will give us that the probability will be 1/4 for all of them. But that's not it. Since the exercise is asking, as you could see in the question, of producing the genotype. What is the probability of producing the genotype from all of these in this cross? We need to consider all of the probabilities that we got before in the Punnett square of each of them. So to getting dominant for AA, it's 1/4. For BB, it's 1/4, and CC, it's 1/4. So we need to multiply 1/4, multiply by 1/4, and 1/4, which will give us 1/64. So, second question. How many unique gametes could be produced through independent assortment by an organism with the genotype AA, BB, and CC? As you can see here, AA is a heterozygous, BB heterozygous 2, and CC is dominant, completely dominant, homozygous. So, now the explanation. So here's the second exercise, where it says identify heterozygous and homozygous genes. In this case, we have this gene that tells us that this one over here, CC, is homozygous, which says that there is only one apportation that this gene can provide to the new gamete. And here we have 1 and 1. One dominant and one recessive chance, which means that this gene can apport to the new gamete. The same with this one, because there is one dominant and one recessive. So one dominant and one recessive. So in this case, as you can see, here there are two options that will provide to the new gamete and two another options. So in this case, since in A dominant there is A recessive, there could be the support from A dominant or A recessive, there are two options. The same with B dominant and B recessive, where there will be two options. But in the case of the double C, there will be just one option, which will not count because it will not provide to the new gamete creation. So in this case, the total sum of the probabilities of this one and this one, of these two genes, will give us that there are 4 options. So third question, tallness T is Snapdragon is dominant to dwarfness. This is a recessive trait, while red dominant flower color is dominant to white recessive. The heterozygous condition results in pink, which means that the combination of a dominant trait and a recessive trait for the color of the flower, it will be represented as pink. A dwarf red snapdragon is crossed with a plant homologous for tallness and white flowers. What are the genotypes and phenotype of the first generation individuals? So first, as you can see, they are asking for the genotypes and the phenotypes for the first generation individuals. That's what we need to get, and with that, first we need the data. Always get the data will make it way easier to understand the problem. So at first, we can see that the dominant traits will be represented with a capital letter, as always. For tallness, it will be T. For the flower color, it will be red. So, in this case, tallness is with a T and dwarfness is with another T, but in this case is a lowercase. Red for a dominant color, and white is a recessive color, so it's a lowercase. But in the case if both of the genes appear, they will share the dominance of the color and get pink. Having said that, we can start identifying the parents. As mentioned before, the first parent will have dwarfness, will be shown by two recessive traits with red, which means that there are two dominant traits in this part of the colors. The second parent, it will be tall, so that's why the 2 T's, and then it will be white, so it will look like this. Then we need to make the crossing over, but as you can notice here, there is not much variation, as we said in another example before. Why? Because both of them are recessive and dominant. So there is only one possibility to get one type of gamete, which will be, in this case, lowercase t and r, or in this other case, it will be capital T and R. Or if you want to say it more properly in biology terms, it will be a dwarfness red and a tallness white. So now we need to get the answer, which will be crossing both of the parents. As you can see here, since there are basically just one type of possible gamete, we can do this type of Punnett square where we only multiply one trait of the parent, because if we recreate the table bigger, it will be the same answer. So first multiply the t's, get the t's in order always. So the first parent will be giving the dominant trait, then the recessive trait and dominant trait, which will give us, as you can see, this will be the genotype, which says that it's dominant recessive, and the phenotype will be tall, as you can see here, because we have a dominant trait. But it will, in this case, since we have in the data that the combination of red and white, it will give us pink. Since we got this, the answer will be tall and pink. Now for the part 2, that it's 3 response questions. You are a world-famous breeder of purple pepper eaters, or PPE. Tragically, your entire stock perishes in a fire except for 2 individuals with heterozygous for all 3 traits desired. These 3 traits are, and as you can see, since they are saying heterozygous, we know that it will be 1 dominant and 1 recessive. Here is the table to identify all of the alleles that we need. For example, in this case, we know that the 2 individuals that survived will be 2-eyed, in this case here, 2 horns, in this case here 2, and no flying since it has 1 dominant and 1 recessive or heterozygous. Now, first question. The traits that bring the big box, or 100 each, are 1-eyed, 1 horn, and flying, purple people eaters. If you breed the remaining 2 purple people eaters, what is the probability that the offspring that has the desired traits? Now we need the data again, which will make us know easier what we need to identify. So the traits that brings the big box will be all of them recessive, and for the ones that are two remaining purple people eaters are these ones, at least as we read before, which will be heterozygous. The first step to get the answer we need to put in the Punnett square, just as the previous exercises. Organize them by the alleles and giving the dominant, recessive in all of the gene to get the gene, sorry. After that, we can answer and build the table where we get completely dominant or homozygous, heterozygous, and a homozygous again, dominant and recessive. The pattern follows again since we have the heterozygous traits, where we have two homozygous and two heterozygous, one is dominant and one is recessive. The same for F, and now we get the probabilities. Since they are asking for all of the alleles to be, all of the genes to be recessive, we can see that it's 1 over the 4 or the table, 1 over the 4 of probabilities and 1 over the 4 answers that we got before. So 1 over 4, 1 over 4, and 1 over 4. And what do? The same as before in the previous exercise, where we multiply the probabilities and we get 1 over 64. So the answer was asking to be written, so this is my type of answer. The separation and random combination of alleles ensure that offsprings inherit one allele for each trait from the parent, allowing the possibility of producing individuals with the desired one-eyed, one-horned, and flying phenotype in 1 out of 64 possibilities. In the end, the cross from the two purple people eaters, it will be 1 over 64 chances to get the offsprings with the desired traits to get the big box. So now I'm going to teach you the chi-square test in Drosophila. You are studying 3 traits in Drosophila or fruit flies. You cross through 2 flies with genotype GGNNRR multiplied by the same genotype and analyze the phenotypes of 512 offsprings. Here is a table that shows the traits, dominant and recessive alleles. In here we can see the body color, which is gray and black. Gray is dominant to the black, which is recessive. Wing shape, which is normal or vestigial, and eye color, which could be either red or brown, depending on the allele that is used and the genotype. So first, to do the chi-square, it's important to write down the null hypothesis and the alternative hypothesis. For the null, there is no significant difference between the expected and the observed group. This is important to remember because the null hypothesis will be always the same for each of the chi-squares. And the alternative will be the opposite. There is a significant difference between the observed and the expected group. So remember, null is no significant difference, and alternative will be always the significant difference between the observed and the expected. After that, we can start classifying the phenotype and genotype. You decide to classify offsprings into 5 categories. Use Mendelian probabilities and the multiplication rule to calculate the expected frequencies, or E, to each phenotype. This will be useful to complete the chi-square later. So here are all the 5 classification of the categories. Gray, normal wings, and red eyes, which are all dominant. Gray, heterozygous, normal wings, homozygous, and brown eyes. Gray, heterozygous, vestigial wings, red eyes, heterozygous. Black, normal wings, heterozygous, red eyes, heterozygous. And black, vestigial wings, and brown eyes. Now, to start with the answer, we need to start with the probabilities. So we use the multiplication rule, which means G multiplied by G and G multiplied by G dominant by G dominant, G recessive by with G dominant, and so on. So after getting that, or if you want, you can do also the Punnett square, but this is easier, it's faster. We can get that 3/4 will be gray because they have the dominant trait and the recessive trait, or they have, for example, or dominant. And in here we have that 1 of the 4 probabilities will be black because they have a recessive trait. We can consider too that there will be 1/2 of the dominant recessive and also a 1/4 of the dominant trait. And as we can see here, since they are the same type of genotypes, by what I mean is that they are heterozygous, 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 we can get the same conclusion for all of these ones, which means 3/4 will be normal and 1/4 will be vestigial, or 3/4 will have red eyes and 1/4 will have brown eyes. Now to continue, as we said before, we need the probabilities. So we can multiply gray normal wings, red eyes for all dominant. Since they are all dominant, they will have the genes, and we can say that 1/4 multiplied by 1/4 and 1/4, just as I explained it in previous exercises, it will be 1/64, and this, then we need to multiply it by 512. As you can see in here, these are the, in this part, these are the offsprings that will be exposed in these exercises. So that's why we're going to multiply them in here. So 1/64 multiplied by 512, you can use your calculator, will give you 8. Same process here, gray heterozygous, normal wings, homozygous, brown eyes. If we check for gray heterozygous, it will give us 1/2, as you can see in this part. Then normal wings, 1/4, normal wings, homologous, that's why it's 1/4, and brown eyes will be 1/4 because it's a recessive trait. 1/32 multiplied by the number of offsprings will give us 16. The same with this one. Gray heterozygous, 1/2. Vestigial wings, 1/4. Red eyes heterozygous, 1.5. It will be 1/16 multiplied by 512, 32. The same, black normal wings heterozygous, 1/4. Red eyes heterozygous, or plus 1/2 and 1/2, which will give us the same. And finally, black, 1/4, vestigial wings, 1/4, and brown eyes, 1/4, or everything multiplied will give us 1/64, which multiplied by the number of offsprings will give us 4. This is the observed data that will be provided always in an exercise. Here with the phenotype, as you can see, the same phenotype as we created before, as we saw before, and the observed group with the values evaluated in the number of offsprings. Now we can create the table. The first part of the table, we have the phenotype, as you saw before, the genotype, that it's basically the crossing over that we did before too. You will have it if you did either the Punnett square, or if you check in here in this process too, as you can see here, we have these parts. So if you did the crossing over, you will be able to get also the how it will look, because in each exercises will be specified how they will look, either all dominant or heterozygous or homozygous. And we need to complete the observed values, which will be provided by basically this table. After that, we can put our values too, which will give us for the first gray normal red, 8, gray normal brown, 16, gray vestigial red, 32, black normal red, 32, and black vestigial brown, 8. Now, after that, we can complete the formula, which means replacing the O, E, and E with numerical values, which should look like this: 200 minus 8/8 squared, 120 minus 16 squared over 16, 80 minus 32 squared over 32, 85 minus 32 squared over 32, and 27 minus 8 squared over 8. After that, we can put this on the calculator, and it will be 4,608, 676, 72, 87.78, and 45.30. After that, we can sum all the values, which means put all of these values in the calculator, and you'll get this, 5,488.9. Taking the degrees of freedom, which means basically counting all of the groups that you have in your problem. In this case, there were 5 groups, minus 1, which give us 4, and the p-value. So to get this, the p-value will always be 0.5. And since we have the degrees of freedom as 4, you'll get 9.49. After this, you can see that the comparison of the values with the p-value, it's huge. It's extremely huge, which means that we can finally get a conclusion since we have all of the data. Our recommendation is to write claim, explanation, and resolution. So in this case, first we need to do the claim, which will be the null hypothesis is rejected and the alternative hypothesis is accepted. Now, the explanation. Since the chi-square value is greater than the critical value from the chi-table square, this shows that the p-value of the observed group has low chances to happen. Because of chi-square exceeds compared to the critical value, this shows that between the expected and the observed groups there is a significant difference. This suggests that the variation cannot be explained by random chance alone, and it is likely due to other biological factors than any chances. This is how you can write down your conclusion in the case that the new hypothesis is rejected. If it's accepted, you need to point out that the alternative hypothesis was accepted and point out why. And this was everything for today. Hope you have a nice day. Thank you.